Hello, class. Welcome to week 7. In some ways, this is starting to get to some of my favorite parts of the class because we now have the background information needed to be able to apply it to be understanding how our cells are working and how our bodies are getting energy. So we're going to start with a brief review of thinking about these catabolic pathways that are breaking down polymers, breaking down carbon-carbon bonds and other bonds into smaller components, into monomers, and getting out energy. And that energy is in the form of ATP. So again, ATP, adenosine triphosphate, it looks like a nucleotide with adenine and that ribose sugar like RNA. But instead of just a single phosphate group, there are 3 phosphate groups. That's the T of a tri-TP. And really what our bodies do is just constantly shuttle between having 3 phosphates and 2 phosphates, letting that high phosphate energy bond be the energy push that many other reactions require. So continuing a little bit of review with photosynthesis and aerobic respiration, we see that the products of one are the reactants of the other, and the reactants of one are the products for the other. And again, what those are organic molecules and oxygen. And in cellular respiration, using the mitochondria, that energy from the carbon-carbon bonds in the organic molecules gets converted to the energy within ATP. In photosynthesis, light energy is used to convert those byproducts, those smaller, simpler, more stable molecules get converted into those more complex, less entropy, organic molecules. So again, we're putting this in connection with the laws of thermodynamics. So again, also at each one of these, some of this energy is going to get lost to heat. So overall, though, we're going to be focusing on cellular respiration. So you need to know cellular respiration. You're starting with glucose, 6 carbons, 12 hydrogens, 6 oxygens. It's a carbohydrate, of course, so 1 to 2 to 1 carbon to hydrogen to oxygen ratio. It's going to involve oxygen, 6 molecules of oxygen. And what we're going to break down is we're going to break down these carbon-carbon bonds, and we're going to release those carbons as carbon dioxide and water. And in doing so, we are going to convert ADP with a phosphate to make ATP. That is the point of cellular respiration. So now we get to think a little bit about how this reaction occurs. And we're going to be talking about energy and how we're getting energy out of these bonds. So the production of water is actually an explosive reaction. Energy is released. It's an exergonic reaction. And you can actually show a little video of what can happen. It'll go boom real quick. Now, do you think that's what's happening in our cells? Of course not. So we're going to see the whole point of cellular respiration is to take that energy and instead of letting it go in one big kaboom, to harvest it in these little incremental steps to get the most energy of it we can, to get the most energy out. And so that's really describing cellular respiration. So a key part of that controlled steps are going to be examples of redox reactions. So we were introduced to this a little bit earlier in the chemistry section of thinking about the terms oxidation and reduction. So oxidation is the loss of electrons. Reduction is a gain of electrons. So some convenient mnemonic devices is oil rig, oxidation is loss, reduction is gain, or Leo says gur, thinking about a little tiger or lion, loss of an electron is an oxidation, gain of an electron is reduction. There's no, like, you just have to remind yourself a bunch and then think about what that means. So sodium becoming a sodium ion is going to lose an electron. That's oxidation. Chlorine gaining an electron will be reduced. So that can happen when you're physically moving an electron, but some of them don't directly involve ions, but rather covalent bonds. And in that case, oxidation, not surprisingly, is going to involve the addition of oxygens. Reduction is going to add the addition of hydrogens. So in this case, methyl is the reducing agent, and because it becomes oxidized. Oxygen is an oxidizing agent because it becomes reduced. It's a flip side where if you're reducing something, you're going to be oxidized. So you need to understand those terms and what they mean, and physically what they mean. So it can be a physical loss of an electron, or you can think about adding a hydrogen is the same as gaining an electron. Because again, hydrogen is simply that single electron and single proton. So what we're going to be seeing today is how cellular respiration takes glucose, takes sugars, and oxidizes them to become carbon dioxides. And in doing so, reduces oxygen to become water. And in the process, produces energy. Okay, so here I want you to try to answer all these questions. And I'm going to assume that you're going to pause right now, and then I will go over the answers. So pause it and answer these questions. So now the answers. So first, identify the molecules. Well, this we can clearly see is glucose. So what are we making up? What are these molecules? Well, this should look familiar. It's water with oxygen and 2 hydrogens. And here we have carbon dioxide, carbon with those 2 blue oxygens. Similarly over here, we have water and carbon dioxide. So which area of the diagram represents you eating and processing food? Eating, that's taking in glucose molecules and breaking them down to get out that energy. So that's going to be the right half. Which half represents a process that plants do? That's photosynthesis. That's taking water and carbon dioxide and making glucose. Which half needs sunlight? Photosynthesis. Which half shows energy being released? That's the right half. That's what is happening in cellular respiration. Which half shows a molecule being built? We're showing glucose being built on the side of photosynthesis. So again, we can think about these systems, but you want to be able to visualize these molecules and think about where is their low energy and where is their high energy. And reminding ourselves as each of these reactions is taking place, a little bit of energy is going to be lost due to that second law of thermodynamics as heat. What we're talking about for cellular respiration, we're going to see is going to be that breakdown of glucose into carbon dioxide and water. And we're going to see that several steps are going to occur. So those 4 steps are glycolysis, a transition, the citric acid cycle, and then oxidative phosphorylation. So this pictures, you need to understand these, and I'm going to spend the rest of this time going over this in depth. Where we have the first step of glycolysis. We're going to see glycolysis is the breakdown of glucose into pyruvate, and this is going to occur in the cytosol. We're going to see how this is substrate level phosphorylation to produce a little bit of ATP. But ultimately, we're going to get some electrons that have high energies in the form of NADH. So that's step 1, again, with more detail to come. Step 2 and 3, we're going to see is the pyruvate oxidation and the formation of acetyl-CoA, and then into the citric acid cycle. Now, these 2 steps are occurring within the mitochondria. So this is where the mitochondria, that organelle, is going to be required. And in these steps, we're going to get back more high energy electrons that we're going to see for that 4th and final step. We're going to get a little bit of ATP out in, again, substrate level phosphorylation. But where we're going to get most of the energy out in the form of the most molecules of ATP is in the 4th and final step of oxidative phosphorylation. We're going to see how that's going to involve the electron transport chain and chemiosmosis. So this level you absolutely must know. In fact, you're going to need to know it in much greater depth than just this. So we are going to be comparing 2 different ways we are going to get energy out of our systems. We're going to have substrate level phosphorylation and oxidative phosphorylation. In substrate level phosphorylation, it's kind of a very simple thing to think about. You have a substrate that has phosphate group on it, and it's going to transfer that phosphate onto that ADP molecule, turning it into ATP. This is going to require an enzyme, a protein that is folded just right, that fits just this specific substrate. and we're going to get that ATP out. I highly recommend you watch this video. Highly recommend it. Okay, so now we're going to get into the nitty-gritty. The first step is glycolysis. Glycolysis is the easiest one to remember because the word itself tells you what you need to know. Glycolysis is glucolysis. Lysis as in when the cell is in a hypotonic solution and it's going to split or lyse. When we talked about hydrolysis and water splitting, glycolysis is glucose splitting. How is it going to split? It's going to go from a glucose molecule with 6 carbons to 2 pyruvate molecules, which each have 3 carbons. What you need to understand is this is a multi-step process. And the very beginning step actually requires energy. So for glycolysis to occur, in order to break down the 6-carbon sugar into 2 3-carbon pyruvates, you need to invest 2 ATP. But there's a second phase where you're actually going to get 4 ATP out. So there is going to be a net production of 2 ATP, because you invested 2, you get 4 out, net 2 ATP formed in one round of glycolysis, breaking a single glucose. You are also going to get 2 NADH molecules during this splitting. So a different graphic showing the same thing, and this is again the level at which you need to understand glycolysis. In glycolysis, glucose is broken up into 2 pyruvate, and in doing so, 2 ATP are used, 4 are formed, net production of 2 ATP, and you produce 2 NADH molecules. So you don't need to know glycolysis at this level. But I am going to point out one particular enzyme that I'm going to point out at the end, which is phosphofructokinase. So again, this is a series of metabolic events where there's going to be a specific enzyme that is going to convert each reactant into a different product, and each reactant or each substrate is going to have a different enzyme. And one of these key enzymes is phosphofructokinase. So this is glycolysis. Again, I don't expect you to memorize it at this level. You do not need to know each of the enzymes or what the shape of the molecules are. But you do need to know what's coming in and what's coming out. So I've said now a few times that 2 NADH molecules are produced in glycolysis. So what is NAD? NAD is an example of what we're going to see as an electron carrier. What we see is NAD is positive, meaning it doesn't have an electron, or you can add a hydrogen and it can be the reduced form containing that high energy electron. So we see NAD switches back and forth depending on where we are within cellular respiration, and it's shuttling these electrons. So it's acting like just a carrier for these electrons. It's a place to put it. Think of it as a taxi cab to carry those hydrogens, which are really carrying electrons. And you can see that in this form, where you have nicotinamide adenine dinucleotide. You can see that it's 2 nucleotides, 2 phosphates, one with adenine, one with nicotinamide. And you can actually see how that NAD will gain that hydrogen and as a result become reduced because it's carrying that extra hydrogen. So it's that reduced form. So now we can start to continue on following these electrons. So aerobic respiration, we just saw glycolysis will happen in the cytosol. But where does aerobic respiration take place? That is in the mitochondria. And again, I want you to be able to recognize them with different types of pictures pointing out different things going on. Where in this cell, you can see the nucleus, you can see the rough ER, you can see those proteins going to the Golgi. And right here, you can see the mitochondria. So we're going to be focusing in on the physical aspects of the mitochondria with its 2 membranes. So you need to know the structure of the mitochondria. It's 2 membranes with an intermembrane space. So there's a space between these. And in the space in the inside, within the inner membrane, that's called the matrix. That's called the matrix. So we can see again the cytosol. The cytosol, that's where glycolysis is going to take place. You have the matrix inside the mitochondria, and then you have the inner membrane, which is that second membrane. So in glycolysis, we take that 6-carbon sugar with its high energy electrons, and we break down glucose 6-carbon into 2 3-carbon pyruvates, releasing 2 ATP and producing 2 NADH molecules. FADH, we're going to see, is also an electron carrier. And we're going to see those are the products that are then going to go into the mitochondria. So the second step is what we're going to see following that pyruvate. It's the pyruvate oxidation. And we're going to see how in the presence of oxygen, pyruvate cleaves off one of the carbons in the 3-carbon molecule to release the first carbon dioxide molecule and forms a complex with coenzyme A. In doing so, another electron carrier is produced. Another NADH molecule is produced. So this is our step 2, the oxidation of pyruvate into acetyl-CoA. So again, step 2, pyruvic acid, 3-carbon molecules, becomes a 2-carbon molecule that enters the Krebs cycle, a carbon dioxide molecule that gets breathed out, and that energy electron carrier. So now we're ready for step 3, the citric acid cycle, which is often also called the Krebs cycle. So the Krebs cycle, you need to know its place, you need to know where it is happening. It is happening within the matrix of the mitochondria, and we are going to see how pyruvate in the form of acetyl-CoA is going to be generating a little bit of energy and more of these electron carriers. So we have step 2, that pyruvate oxidation, and then the step 3, this is the level I expect you to know the citric acid cycle. I expect you to know that it's a cycle, meaning it is continuously going and going as long as you are shuttling in acetyl-CoA, and that you have enough NAD+ to take those electrons in the form of a hydrogen. So the citric acid cycle is going to produce 3 molecules of NADH, 1 molecule of FADH2, so again, carrying those electrons, and you're going to get a little bit of energy out, again, through that substrate level phosphorylation. Now, you started step 2 with 3 carbons. In step 2, you lost 1 to carbon dioxide. You lose the other 2 carbons, breaking down that glucose into the other 2 molecules of carbon dioxide that are released within aerobic respiration. This is again within the citric acid cycle. You need to know the citric acid cycle, aka the Krebs cycle, at this level. You do not need to know it at this level where we're actually going through the different molecules. Equally, you do not need to understand what is happening at this level knowing these different enzymes. But I did want to continue to have this on the slide so you understand what physical reactions are taking place. How do these metabolic pathways convert the different products into this cycle that can keep going and going and going? So you need to be able to make sense of this, but you do not need to have this part memorized. So again, the citric acid cycle, we can think of it where we're taking that acetyl-CoA, it is entering the citric acid cycle, it is being broken down into carbon dioxide, and in doing so, taking those electrons and shuttling them on that NADH and FADH2. The carbon dioxide leaves. The electrons get carried to the final step, which is the oxidative phosphorylation. Now, this is going to occur within a very specific part of the mitochondria. And this is the final step where we're going to get out most of that energy, where most of that ATP is going to be generated. And how is that ATP generated? That ATP is generated as the energy from those electrons being carried by that NADH and FADH2. And in doing so, we're going to see there's a little passing of that energy, passing of the electrons, taking a little bit and stepping down. And we're going to see that's what's happening in the electron transport chain, where you start with high energy in electrons, and they end up having low energy electrons at the end, which is finally going to get accepted by oxygen to make water. Each of these steps is an example of a redox reaction that we talked about at the very beginning. That shuttling of electrons, redox reactions, reduction, and oxidation. And we're going to be able to get that energy from the electrons and convert it into ATP. How this is happening? So we're going to see the electron transport chain, or the ETS, is going to take those high energy electrons from the carriers FADH2 and NADH. It's going to produce back NAD plus and FAD, and it's going to shuttle those electrons through the electron transport chain, which is literally in the inner mitochondrial membrane. So how does the energy from high energy electrons get converted into that chemical energy in ATP? That's the fourth stage of aerobic respiration. So this is the electron transfer chain and the electron transfer phosphorylation, where we have those high energy carriers that are depositing their electrons. They get shuttled in a series of redox reactions until that electron finally needs a place to go. Where does it go? It finds what's available, which is an oxygen. It finds a hydrogen ion that's also available, and that electron combined with those oxygen and hydrogens forms a water molecule. Now, as that electron is being shuttled from one protein to another down the electron transfer chain, hydrogen ions end up getting pulled through. I sort of like to think of it as tricking the hydrogen ions. They get sort of tricked by following that negative electron to go through the membrane when they normally could not, and in doing so get stuck in this inner membrane space. They then want to go back down their concentration gradient, but the only way to do that is to find that particular door, that particular protein that's going to let them flow back through that inner mitochondrial membrane. That protein is called ATP synthetase. Why? Because it is synthesizing ATP from ADP and a phosphate group. And how is it getting the energy? It's literally the force of these hydrogens through this channel that turns this crank. So you can see it's literally turning a crank as the hydrogens flow through, and it turns this little rod. And as a result, that phosphate group forms a bond with ADP to form ATP. You can see a little video. You have ADP, the hydrogen ions are flowing back through, and that produces ATP. So going through this again, reviewing the key steps, it's a bit tricky. The electron transport chain and chemiosmosis. What happens is those energy carriers, NADH and FADH2, deposit their electrons, which get shuttled from one protein to another, losing their energy ultimately until it needs to get deposited onto oxygen, forming water. When it was doing that change, it made those hydrogen ions, tricking them into that inner membrane space. Ultimately, those hydrogen ions are going to go, based on diffusion, down their concentration gradient through the only hole available, which is ATP synthetase, and in doing so, turn this crank to force that phosphate group onto ADP, producing ATP. It's called chemiosmosis because it's that ion moving down its concentration gradient. So you should now be able to label all the parts of this figure. And I'll go through it, so pause it and see if you can answer. See if you can identify these hydrogen ions. See if you can identify that NADH and NAD plus are going to shuttle and bring those electrons in. FADH2 is going to go back to FAD, and in doing so, shuttle that electron. Those electrons are going to continue to be forcing those hydrogen ions through the inner membrane space, where ultimately they're going to build up and go back down through ATP synthetase. Ultimately, that electron needs to be deposited somewhere. It's going to be deposited on hydrogen and oxygen. So again, you have this slide explaining what is going on, thinking about that electron transport chain and that chemiosmosis of hydrogen moving down its concentration gradient. Oxidative phosphorylation is the term for both of these processes. So again, cellular respiration overview starts with glycolysis. We get a little bit of energy. The transition state and the Krebs cycle, we get a little bit of energy. We get most of our energy in that final step, the electron transfer chain and oxidative phosphorylation. So if we want to calculate how much energy we get a total of 36 molecules if you add up how we actually get it. which if you break down how many energy molecules are actually in one molecule of glucose, we only get about 40% of the energy back. And that might sound really terrible until you look at what's the efficiency of vehicles, and we find out it's only about 20%. And in your reading, you'll see that it really is remarkable how our body uses ATP, a teeny tiny amount of it, but we're constantly just shuttling on and off of that last phosphate group. So in a day, we use about 3 to 5 pounds, but we only actually have 1/5 of an ounce of it. So we're constantly recycling. So I'm going to end this video where I'll start the next video, which is just reminding us what we just saw was aerobic respiration, where we take pyruvate, convert it to acetyl-CoA, enter the Krebs cycle, and in that final electron transfer state, use oxygen to get ATP out. But what happens when there is no oxygen? There is no oxygen, there is no electron transport chain, there's no Krebs cycle, there's no transition. You're just left with pyruvate. And the second video, I'll go through fermentation and anaerobic respiration.